Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Kerala SET Biotechnology question paper January 2023. I have solved the paper with explanations. This is part 2 of the discussion. You can all I have also uploaded previous year Kerala SET Biotechnology question papers. You can view my channel playlist for the same. If you are new to my channel, please don't forget to subscribe it. So coming to the next question. Luciferase genes are used as reporter genes for screening. Choose the correct statement for them. They are obtained from fireflies only. No. Not only fireflies, but also they are obtained from bioluminescent bacteria and a sea pansy. Then, the detection requires the provision of substrate which produces light. That is correct. Enzymes such as beta-galactosidase require substrate X-gluc to produce light. It is not X-gluc, it requires X-gal to produce blue light. Luciferase genes are preferred over fluorescent protein. No. Fluorescent proteins are more preferred over the luciferase genes because they don't require substrates like the luciferase genes. So your correct answer is option B. The next one. Sometimes successive rounds of screening of a genomic library are carried out and an ordered collection of clones is done in a linear fashion. Then the process is called as chromosome walking. It is the process of successive rounds of screening of a genomic library and it leads to the collection of clones in a linear order. That is called chromosome walking. The next one. Active transcription of GAL1 and GAL10 promoters is suppressed in the presence of. So GAL genes are repressed by glucose but activated by or induced by galactose. So they are suppressed by glucose. So the answer is option A. Then next one. The polymerase used in PCR based mutagenesis. It is PFU polymerase. Why is it used? Because it is used to ensure blunt ends and avoid second site mutations. To avoid that, PFU polymerase is used. What is the holiday junction? The site of strand break? No. The site of heteroduplex DNA formation? No. Formation of a crossover complex? Yes. So answer is option C. The next one. Which one is a human amnion derived cell line? HeLa cells. HeLa cell lines are derived from cervical cancer cells of Henrietta Lacks. So it is not HeLa cells. Now wish. Yes, it is a human amnion derived cell line. So B is the answer. What is L? L cell is fibroblast cell line that was isolated from the areolar adipose tissue of a 100-day-old male mouse. What is MRC5? These cells are derived from embryonic lung tissue of 14-week-old Caucasian male fetus in 1966. Then next one. Which of the following statement is incorrect for culture and maintenance of mammalian cells in vitro? Transformed cell lines need an exogenous supply of serum for growth. Yes, that is correct. The cells that are obtained directly from an organism is primary culture. Yes. Trypsin is added to cell culture dish to maintain cell viability and health. That is not correct. Because trypsin is added to detach cells and also to separate cells from each other. So they can be transferred to a different vessel for replating. That is why you add trypsin. So 71C is the answer. They have asked which is the incorrect statement. Then HEPS is generally used for pH balance in animal cell culture medium. That is also correct. Then if log 4 1 by 4 is equal to x, then x is. So log 4 4 raised to minus 1. 1 by 4 is 4 raised to minus 1 is equal to x. Minus 1 log 4 4 is equal to x. X is equal to minus 1. Answer is option A. The next one. All the statements regarding RFLP and RAPD are true except. RAPD is quick method compared to RFLP. Yes, that is correct. RFLP is more reliable than RAPD. Yes. Species specific primers are required for RAPD. RAPD requires random primers, not species specific primers. So C is the answer. Radioactive probes are not required in RAPD. That is also correct. The next one. Which among the following gives the best outcome for the creation of transgenic animals when DNA is micro-injected to fertilized egg? After the fusion of male and female pronuclei, before the fusion. Yes, that is correct. Before the fusion of male and female pronuclei. See, what is the procedure of microinjection? Using a fine hollow needle, a solution of DNA containing the transgene is injected into the male pronucleus of the fertilized egg before it fuses with the female pronucleus. Okay, so here they have asked which among the following gives the best outcome for the creation of transgenic animal when DNA is micro-injected to fertilized egg. So before the fusion of male and female pronuclei, that will give the best outcome. Then next one. In plant tissue culture system, what is the function of elicitors? Induce cell division and callus formation. Stimulate production of secondary metabolites. Stimulate production of secondary metabolites. Answer is B. Then next one. During recombinant insulin synthesis, the bond between insulin polypeptide and galactosidase can be removed by using. So what are the steps in insulin production? First, DNA was made based on the protein sequence of the two insulin chains, that is A and B. Then you got a single stranded DNA chain. Then you have to convert it into double stranded DNA chains using DNA polymerase. Then you have to insert these into plasmid. It is inserted next to the beta galactosidase gene. So one in the expression vector you have got a tetracycline resistance gene for selection. So B chain is also similarly inserted next to beta galactosidase gene in the plasmid. Upon expression you get a fusion protein, that is beta gal attached to A chain and beta gal attached to B chain. Then you do CNBR treatment. So CNBR treatment, the beta galactosidase part of the protein is cleaved off and discarded by CNBR is cyanogen bromide. Then you isolate and purify the A and the B chain, combine them, reduce and reoxidize, you get the disulfide bonds form and the usual human insulin has been made in the bacteria. So they have asked the bond between insulin polypeptide and galactosidase can be removed by cyanogen bromide. Answer is option A. Then next one. Freedom 2, the first virus-resistant transgenic crop against cucumber mosaic virus, is a transgenic squash variety commercialized in 1995. It carries gene coding for. So Freedom 2, which is a virus-resistant transgenic crop, it carries genes coding for. So it carries gene coding for the virus coat protein genes. So answer is option C. The G protein coupled receptor rhodopsin is composed of a transmembrane apoprotein opsin and 11 cis retinal. Answer is A. The next one. The region of electromagnetic spectrum used in NMR to promote resonances. It is radio frequency. All these are direct questions. That's why there is nothing much to explain there. The unit of absorbance which can be derived from Beer-Lambert's law. Absorbance doesn't have a unit. It is unitless. But molar absorptivity has units, that is liter mole raised to -1 centimeter raised to -1. So the unit of absorbance which can be derived from Beer-Lambert law is no unit. Answer is option D. Then next one. The centrifugation technique is used to separate mitochondria from other organelles in the cell extract. So how will you separate mitochondria from other cell organelles? That is by differential centrifugation. Answer is option B. The half-life of a radioisotope is half of the time taken for complete decay, half the time taken for half the decay, time taken for complete decay, time taken for half the decay. Answer is option D, that is time taken for half the radioisotope to get decayed. Then next one. The part of the compound microscope which helps in gathering and focusing light rays on the specimen to be viewed. So it is the condenser lens. Answer is option A. Then next one. The respiratory chain of bacteria is associated with the. So it is associated with the mesosome. Answer is option A. Chrysolaminarin is the reserve food of. It is the reserve food of Bacillariophyta. Option A. Which of the following is a family of bacteriophage lambda? It is Cyphowiridae. Answer is option A. What is the mechanism of action of polymyxins for inhibiting growth of microbes? Inhibition of cell wall synthesis? No. Polymyxins don't do inhibition of cell wall synthesis. Disrupting cytoplasmic membrane? Yes, that is correct. Polymyxin disrupts cytoplasmic membrane. Inhibition of nucleic acid and protein synthesis, inhibition of specific catalytic enzyme. That is also not correct. So B is the correct answer. The principal function of class 1 and class 2 major histocompatibility complexes protein is 2. So these classes, they present antigen to T cell antigen receptor. Mediate immunoglobulin class switching? No. Stimulate the production of interleukins? No. Transduce the signal into T cell following antigen binding? No. Present antigen for recognition by T cell antigen receptor? Yes, that is correct. They are meant for presenting of the antigen. So 87 D is the correct answer. Then next one. Which of the following is a major focus of the technique of tissue engineering called autologous chondrocyte transplantation? So in ACT or autologous chondrocyte transplantation, chondrocytes are collected, multiplied in the laboratory to increase cell number and then re-implanted with a special surgical technique into the damaged area of the knee. So, what is the major focus of the technique of tissue engineering called autologous chondrocyte transplantation? Chondrocyte are cartilaginous cells. So, the answer here is option A, that is cartilage. Name the part of the processed antigen that bind to the MHC molecule and recognized by T cell. It is the agritope. Answer is option B. Identify the second generation thrombolytic drug from the following drugs. So second generation thrombolytic drug is alteplase. Answer is option B. The radioactive element in which is used in radioimmunoassay: tritium, carbon 14, iodine 125. All these are used in radioimmunoassay. So answer is option D. Then next one, which of the following do not incorporate in the genome and replicates in the cytoplasm? So out of these, pox virus does not incorporate in the genome and replicates in the cytoplasm. Answer is option A. Which of the following is not considered as a functional food? Probiotics, dietary fiber, pharmaceuticals, omega-3, PUFA. So out of these, which is not considered functional food? Pharmaceuticals is not considered functional food. Answer is option C. The next one. Identify the non-essential amino acids which can be synthesized in the body. So they are tyrosine and glutamine. So 3 and 4 only. The answer is option C. K. Itakura and collaborators chemically synthesized DNA sequence for the two insulin chains A and B. These were incorporated in two separate PBR322 vectors and peptide chains were synthesized, then combined in vitro to produce active insulin in the year. So when was this recombinant insulin prepared? In which year did K. Itakura and collaborators conducted the experiment? So it was in option C, that is 1977. Various measures taken to prevent any risks to normal organism from transgenic organism are known as biosafety. Answer is option A. Then next one. After cleaving the sequence, the nature of the ends created by type 2 endonucleases is. So type 2 endonucleases, they create cohesive ends. So either the ends are single stranded or they are double stranded. So answer is option C. Sometimes blunt ends are produced, sometimes cohesive ends are produced by type 2 endonucleases. Then next one. Choose the correct statement for the infectious particle of a lambda phage. It is single-stranded genome. It is a circular double-stranded genome. The ends are blunt with cos sequences. The ends are created by cleavage at cos sites during phage packaging. That is more correct. So 98D is the answer. What do you mean by extracorporeal medical procedure in tissue engineering? Extracorporeal means which is performed outside the body. So 99 answer is option B, outside of the body. The various steps for construction of libraries are fragmentation of DNA, isolation of genomic DNA, amplification, ligation and introduction into host vector preparation. The correct order of construction of libraries in order of start to end is. So first comes isolation of genomic DNA, then comes fragmentation of the DNA, then comes vector preparation, then ligation and introduction into the host, then amplification. So the answer here is option B, 2145543. Then choose the incorrect statement for oligo-DT cellulose. It is used for separation of polyadenylated mRNA from other species of RNA. Yes, that is correct. That is why you use oligo-DT cellulose. So polyadenylated means it will have A at the ends, which will bind to the T in the oligo-DT cellulose, and it will be bound to the stationary oligo-DT cellulose, and the other species of RNA will be eluted out first, and then only later you can elute your mRNA alone, which is bound to oligo-DT cellulose. Oligo-DT are covalently attached to the solid support via OH bonds. Yes. A solution containing RNA is passed through the column. Yes. Poly A tails attached to the oligo-DT by ionic bonds. It is not ionic bonds, by hydrogen bonds. A hydrogen bonds with T through two hydrogen bonds are formed between A and T. Then next one. The chromosomal alteration which causes retinoblastoma. It is deletion in chromosome 13. Answer is option C. Then next one. Name the chemical carcinogen which causes prostate cancer. It is cadmium. In myasthenia gravis, a long-term neuromuscular junction disease, dash autoantibodies are positive. So myasthenia gravis, it is acquired autoimmune disease in which autoantibodies against acetylcholine receptor at the neuromuscular junction are produced. So autoantibodies are positive for autoacetylcholine receptor. Then next one. The first recombinant vaccine approved for human use targeted the surface antigen of hepatitis B virus. A question based on hepatitis B virus vaccine have been asked in July this same year in the SET Biotechnology exam. The type of biosensor used for therapeutic applications. So it is nano-based biosensor that is used in therapeutic applications. The drug which is used to isolate hybridoma cells from the media by blocking the de novo pathway for nucleotide biosynthesis. Everybody knows hybridoma technology, so I am not going to explain much about that. It is aminopterin. So option C is the answer. Answer is option C. Then next one. The most commonly used monoclonal antibody for treatment of breast cancer is Herceptin. That is the monoclonal antibody used in the treatment of breast cancer. In the two-compartment model of pharmacokinetic analysis, name the organ that make up the peripheral compartment. So out of these options, kidney, liver, lungs come under central compartment and pancreas come under peripheral compartment. So they have asked name the organ that make up the peripheral compartment. So it is pancreas. Which of these absorption methods involves engulfing of the extracellular drug? So endocytosis is the answer. Passive diffusion no. Facilitated diffusion also no. It is just diffusion. Ion pair transport is also not correct. Then next one. Which of the following is the most common variation in the human genome? So most common variation is single nucleotide polymorphisms. Answer is option D. CYP2D6 polymorphism can affect. So CYP2D6 enzyme, it is an enzyme that catalyzes the metabolism of a large number of clinically important drugs such as antidepressants, neuroleptics, antiarrhythmics, lipophilic beta adrenoceptor blockers and opioids. So for those drugs that are metabolized by CYP2D6 to inactive metabolites, CYP2D6 inhibitors may result in toxicity. So CYP2D6 polymorphism can affect toxicity. Answer is option C. The drug which is not classified in the criteria based on the pharmacological effect. So pharmacological effects of a drug provide a wide range of drugs that have the same effect on the specific type of problem. Analgesics have pain-killing effect, antiseptics prevent growth of microbes, and antipyretics help in lowering body temperature and fever. But antihistamines, these drugs interfere only with the natural action of the histamine. So the answer here is it is not classified in the criteria based on pharmacological effect is antihistamines. Answer is option A. ADME in pharmacokinetics means it means absorption, distribution, metabolism and excretion. Answer is option C. Then next one. The term used to signify a preparation that appears identical to the preparation of an active drug but which has no biological activity. So it is identical to the preparation of the active drug but it doesn't have any biological activity. That term is called as placebo. Answer is option B. Which of the following is one of the rules in Lipinski's rule of 5? Lipinski rule to evaluate drug likeliness or determine if a chemical compound with certain pharmacological or biological activity has chemical properties and physical properties that would make it an orally active drug in humans. For that only Lipinski rule is followed. That is to evaluate whether the drug could be given orally to humans, make it an orally active drug to humans. For that only Lipinski's rule is followed. So what are the components of the rule? No more than 5 hydrogen bond donors. No more than 10 hydrogen bond acceptors. A molecular mass less than 500 daltons. A calculated octanol water partition coefficient that does not exceed 5, that is log P. So, what are the statements that they have given in the options? A molecular weight equal to 500. They have told molecular mass less than 500 Dalton. So, A is not correct. No more than 5 hydrogen bond acceptor groups. It is no more than 10 hydrogen acceptor groups. No more than 10 hydrogen bond donor groups. It is said no more than 5 hydrogen bond donors. So B and C are also not correct. A calculated log P value less than 5. Yes, what is log P? It is octanol water partition coefficient. And its value, it shouldn't exceed 5. So D is the correct answer. Then next one. Which of the following statements best describes an induced fit? Process by which a binding site alters the shape. So binding site is altering the shape such that it is ready to accept the drug. That is not so. That is not induced fit. The process by which drug adopts the correct binding conformation before entering the binding site. That is also not correct. The process by which binding of a drug to a binding site alters the shape of the binding site. Yes, that is induced fit. Answer is C. The process by which a binding site alters the shape of the drug into the binding conformation before binding. So that is also not correct. It is the process by which binding of a drug to a binding site alters shape of the binding site. Then find the next number in the sequence. So how they have done was 1 square plus 2 square is equal to 5, 1 square plus 2 square plus 3 square, then 4 square, then 5 square, then 6 square, you get 91. So the answer here is option A, that is 91. The microorganism used for the production of recombinant humulin by Sanofi and Eli Lilly is, it is Escherichia coli. Which among the following drugs inhibit platelet aggregation by inhibiting the enzyme cyclooxygenase and formation of thromboxane A2? It is aspirin. So answer is option A. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, don't forget to subscribe. Then I have also uploaded previous years Kerala SET Biotechnology question papers. You can find it in my channel playlist. Thank you.